I think with that, we'll move on to the next presentation, which will be by Hassam El Sayed. Hassam has been working in the lab, in particular, looking at transcriptomics. And so we have data now from a number of these kinds of studies, but this is related specifically to Nocardioform placentitis. So Hassam?
Hello, everyone. I'm very glad to share with you what we have learned from the transcriptomic analysis of the chorioallantois from mares with Nocardioform placentitis. As most of you know, Nocardioform placentitis is associated with three major events. The first event is placental inflammation, which is characterized by a focal mucoid inflammation in the base of the gravid horn or in the horn. And this placental inflammation will lead to the second event, which is placental separation. And in this transabdominal ultrasonography, you can see the separation between the uterine wall or the endometrium and the chorioallantois and the accumulation of echogenic fluid. Both placental inflammation and the placental separation will lead to placental insufficiency, which in turn will passively affect the fetal growth and the pregnancy outcome. And taking a look, looking at the PubMed timeline over the last 30 years, you can find about 14 publications about Nocardioform placentitis. Most of these publications focused on the etiology and the epidemiology of the disease, which is important for us to know. But unfortunately, none of these publications focused on the molecular mechanisms or the pathways associated with Nocardioform placentitis. And this generates a huge gap in our knowledge about the disease. And maybe it is limiting the advancement toward efficient diagnostic tools and the treatment protocols for the disease. So how to fill these gaps in our knowledge? In this case, we need a rapid and a holistic view of all the molecular mechanisms taking place. And one of the ways to answer these many questions is to apply omic technologies. And the omic technologies include many technologies such as genomics, which is looking to all the genes in a specific tissue, transcriptomics, which is looking to all the transcript in a specific tissue, proteomics, which is looking to all the proteins in a specific tissue, and the metabolomics, which is looking to all metabolite in a specific tissue. In this study, I will focus on transcriptomics, which means we are using high output RNA sequencing to characterize all the transcript in the placenta. So in 2017, we had the opportunity to collect chorioallantois from mares immediately postpartum that were affected with Nocardioform placentitis, along with control samples. And you can see we have 4 Nocardioform placentitis and 4 control. And in this case, there was no significant difference between the gestational age in both control group and the Nocardioform group. However, there was a significant decline in the full birth weight in the Nocardioform placentitis group in comparison to the control group. Then we collected or we gathered the chorioallantois samples. In this photo from Dr. Rebecca Ruby, you can see the Nocardioform lesion. So we have 2 collection sites from the Nocardioform placentitis. The first sampling site is the boundary or the margin of the lesion, which is the active part of the lesion, as Dr. Ruby pointed out before. So in this area where you can see an elevated chorion and it have red color, so we collected the margin of the lesion and we named this sample the Nocardioform lesion, or NBL. The second sample was collected from the area where the chorion is red and velvet in appearance, and there was no significant or there was no gross pathological changes. And we call this region the Nocardioform normal region, or NBN. In addition to that, we have also 4 samples from normal postpartum chorioallantois, and this is the control group. Then we extracted the RNA from all the chorioallantois samples, and we sequenced them on NextSeq 500. Then we received the raw reads, and we mapped them to the latest equine reference genome, which is ECAP3. We quantified the genes or the transcript using cufflink, and we identified the differentially expressed gene using cuffdiff. So moving to our result, in this heat map, you can see that we identified almost 3,000 differentially expressed genes between the Nocardioform lesion and the Nocardioform normal lesion or control. So here are the differentially expressed gene. The red dots reflect or point to the Nocardioform lesion or NBL. So these are the 4 samples from Nocardioform placentitis lesion. The yellow dots represent those Nocardioform placentitis normal region. The green dots reflect those samples collected from control samples. And the downregulated genes are green. The upregulated genes are red. And here you can see a clear distinction between the Nocardioform lesion and the other samples from control and the normal region. This is reflected on this principal component analysis or BCA plot. And you can see that the red dots or the Nocardioform lesion clustered or grouped together, while the control and the Nocardioform normal sample clustered together in this area. So there is a distinct separation between the Nocardioform lesion and the Nocardioform normal region or the control. Interestingly, within those 3,000 differentially expressed genes, we identified about 79 differentially expressed genes which were exclusively expressed in the lesion. So you can see here this blue color mean this, in this case, it was 0 FPKM or 0 fragment per kilobase of reads. So there was no expression in the control samples for these 79 genes, but these genes were expressed in the, or switched on in the Nocardioform lesion. And these genes are very important because these transcripts are coding for proteins. So if these genes are not expressed or not expressed in the control and only expressed in the lesion, so it might be a possible marker in the biomarker in the future. Maybe we can identify a leakage of this RNA in the blood of the mare, or maybe the protein coded by these transcripts are reflected on the mare circulation. But this needs further evaluation and validation on the serum or plasma samples that we have. So moving back to the total number of differentially expressed genes, which are 3,000 genes, we cannot look one by one between these 3,000 genes. So one of the ways is to find the pathways these genes are involved in. So to answer this question, we run pathway analysis, and we identified that these genes are involved in pathways such as inflammation, pathways such as angiogenesis dysregulation, and apoptosis. And interestingly, these 3 major pathways could explain placental inflammation. So the inflammatory signaling could explain placental inflammation. The angiogenesis dysregulation could explain placental insufficiency. And the apoptosis could explain the placental separation. Then we asked another question. Among those differentially expressed genes, what are the key regulators triggering these pathways? So we run upstream regulator analysis, and we identified 82 upstream regulators. Those upstream regulators included toll-like receptors, such as toll-like receptor 2, transcription factors such as ERF1, ERF7, STAT1, STAT2, STAT4, and the cytokines such as interleukin-1 alpha, interleukin-1 beta, and interleukin-6. So to bring everything together, in this figure, I'm going to summarize the key regulators and the molecular mechanisms which lead to placental inflammation, placental insufficiency, and placental separation during Nocardioform placentitis. So starting from the beginning, so we found that Nocardioform placental infection was associated with upregulation of pattern recognition receptors, such as tool-like receptors and the node-like receptors. And these are the earliest mechanisms of the host to recognize the pathogen. The upregulation of node-like receptors was associated with activation of the inflammasomes, which was reflected by upregulation of caspase-1, interleukin-1 beta, interleukin 18, gasdermin D, with consequent pyroptosis and necroptosis. On the other hand, if we go to the tool-like receptor, the tool-like receptor's activation was associated with upregulation of a wide array of transcription factors, and the upregulation of these transcription factors was associated with upregulation of cytokines, chemokine, diabetes-related molecules, with subsequent increase in the leukocytic infiltration and inflammation. And this inflammatory cascade could explain the placental inflammation. On the right side of the screen, you can see the immunolabeling of calgranulin B or 8. And you can see an extensive immunolabeling of calgranulin B in the Nocardioform lesion in comparison to the control. Calgranulin B is one of the chemokines. So going back to the main figure, so the increase of leukocytic infiltration and inflammation was associated with increase in the matrix metalloproteases, such as matrix metalloprotease 138. And it is well known that matrix metalloproteases are implicated in extracellular matrix degradation. Also, we found there is upregulation of several caspases, and it is well known that these caspases are involved in apoptosis. It is well known in the horse and also in cows that extracellular matrix degradation and apoptosis are essential for placental separation. So the upregulation of matrix metalloproteases and the caspases might explain the placental separation that we see in the cases of the Nocardioform placentitis. On the right side of the screen here, you can see an intensive immunolabeling of caspase 7, which is one of the caspases involved in apoptosis. You can see intensive immunolabeling in the Nocardioform lesion in comparison to the control samples. Additionally, the placental inflammation was associated with angiogenesis dysregulation, as reflected by the downregulation of vascular endothelial growth factor A and its receptor, vascular endothelial growth factor receptor 1 and 2. And on the right side of the screen, you can see the intensive immunolabeling of vascular endothelial growth factor in the normal control placenta and the lower expression of vascular endothelial growth factor A in the lesion in the placenta coming from the Nocardioform lesion. Also, the angiogenesis dysregulation was associated with placental hypoxia, as reflected by the upregulation of hypoxia-inducible factor 1 alpha, which is a biomarker for hypoxia. Also, the placental inflammation was associated with a decrease in the solute carriers, a downregulation of several solute carriers. These solute carriers are very important because these carriers are essential for the transportation of glucose, amino acids, fatty acids, vitamins from the placenta to the fetus. And the downregulation of these solute carriers mean there is a defect in the transportation of these nutrients from the dam side to from the placenta to the fetus. Also, there was a reduction in the IGF signaling or the insulin-like growth factor signaling. And it is well known that insulin-like growth factor is very important for fetal growth. And when you can see reducing its availability in the circulation or in the placenta, also you can see a downregulation of the IGF receptors 1 and 2. So this means there is a reduction in the IGF signaling, which might affect the fetal growth and development. Also, we found a downregulation of several surfactants. It is well known that surfactants are important for the function of the fetal lung. Also, we found there is a downregulation of the progestin, the non-genomic progesterone receptor, or the membrane receptor of progesterone membrane receptor, and this is associated with a decline in the progestin signaling. And the decline, it is well known that progestin are anti-inflammatory, so the decline in the progesterone will increase or make amplification of the placental inflammation. Also, with these placental inflammation, we found that there is a lot of placental regulatory genes, or there is dysregulation in the, there is upregulation of some of the placental regulatory genes, there is downregulation of some of these genes, and these genes are very important for the placental function. So all in all, we can see that the alteration in the placental function, the angiogenesis dysregulation, the placental hypoxia, the decline in the nutrient going to the fetus through the downregulation of the solute carriers, the reduction of the IGF signaling, and the decline in surfactant, and the decrease in progestin function, all of these will passively affect the placental function and will lead to the third event, which is placental insufficiency. In addition to that, we found also that during Nocardioform placentitis, there is upregulation of several antimicrobial peptides, which is a defense mechanism against the invading microorganism. It is important to note this also. And finally, the placental inflammation, the placental insufficiency, the placental separation will passively affect the fetal growth and the pregnancy outcome. Moving from the transcriptome, I have two slides about the microbiology because Dr. Erdal and Dr. Ruby bring a very good point. So there is many cases which associated with mycoid placentitis, but we don't find, or the PCR is usually negative for Amycalotopsis or Coxiella. And as Dr. Ruby mentioned, this may be due to the involvement of other bacteria in the Nocardioform placentitis, or due to the difficulty in detection of the bacteria after antimicrobial treatment. So what we did last year, it is just a preliminary result. We are planning to submit more samples for 16S metagenomic sequencing. So last year we submitted about 10 cases for 16S sequencing. Unfortunately, 6 of them didn't work due to technical error, but we have the result from 4 samples. One is control, and those red squares reflect high abundance of the bacteria. The blue squares reflect low abundance of the bacteria. And if you look to the control sample here, you can see there is a high abundance of a wide diversity of the bacteria. But if you go to the control, you can see this one was, this Nocardioform case was positive for Amycalotopsis. You can see here there is high abundance of Amycalotopsis. The second high abundance in this case was Trichococcus. So there is another bacteria was associated with Amycalotopsis. The second case, there was no bacterial growth, but we found that this case was associated with the growth of different bacteria. One of these bacteria is Cellulomonas, and it is very important to remember that Cellulomonas in 2014 was associated with some Nocardioform placentitis abortion. The third case was a Coxiella equi, and you can see here Coxiella have high abundance in the sequencing, but in addition to Coxiella equi, there is another bacteria associated with it. This have many implication, because if you are targeting one bacteria by the antibacterial drug, maybe the other bacteria which is present is not affected by this antibiotic. So in conclusion,
we need to wrap it up, please.
Sorry. Yes, I will finish now. Sorry. Okay. So finally, we explained the 3 events that we have: the placental separation, placental inflammation, placental insufficiency. And the use of therapeutic inhibition for these key regulators, such as toll-like receptor, might be a promising treatment. There is a lot of toll-like receptor antagonists under investigation now in third phase clinical trials. Also, we found that placentitis is associated with angiogenesis dysregulation, which lead to hypoxia. So maybe the use of medicament which improve angiogenesis and the blood flow might be beneficial for these cases. Some groups have shown that estradiol is important for angiogenesis, so maybe we need to evaluate it. Also, we show those exclusively expressed gene, they might be a biomarker. Also, we show the variety of bacteria which is associated with Amycotopsis and the Coursiella equi in the cases of Nocardioform placentitis. By this, I would like to thank everyone. Sorry for the delay.
Thanks, Hassam. Very nice. I think because we are running behind schedule, we're going to go straight to the next presentation
Sorry for that.
and ask Dr. Puy